Hello everyone, this is my lecture on genetic inheritance or the Mendelian mode of genetic inheritance. So let's begin. First of all, the Mendelian mode of inheritance has four types of genetic inheritance: the autosomal dominant type, the autosomal recessive, X-linked dominant, and X-linked recessive. So let's begin up with the autosomal dominant. Now, the basic features that is mandatory to understand and know about the autosomal dominant inheritance. That is, first is frequency of occurrence in male and female is equal because it's autosomal dominant. It actually, you understand why it is, right? Now, moving on, that can be expressed in heterogeneous state. In heterogeneous state, it can be expressed, and there is no carrier state. There is no carrier state, of course, because the carrier state dies because of the dominant alleles. If it appears in both of the genes, we all know there are two pairs of allele for a particular gene. So if both the alleles carries the particular disease gene, so it can lead to death in autosomal dominant state. So the heterozygous state is the one that actually have the affected autosomal dominant character. Then and the complete type, there won't be any carrier. Yeah. Now that usually it manifested in adulthood. Now the genetic autosomal dominant genetic disorder is inherited in adulthood and manifested in adulthood. The clinical feature appears in adulthood due to the defect in structural protein. Now this is the thing that you have to remember, that autosomal dominant trait is actually occurring due to the defect in the structural protein. Just remember this is important. Now moving on, there are two major properties that you have to remember about the autosomal dominant inheritance. The first one, that is variable expressivity and incomplete penetrance. What is variable expressivity? Variable expressivity means in clinical features, the individuals carrying the autosomal dominant disorder may vary in their clinical manifestation. Their features may vary. They have different sort of features and quality of their manifestation. So that is why they say the variability in the expressivity. The manifestation may vary from person to person. And incomplete penetrance. In incomplete penetrance means not every person will have the clinical feature can be manifested. Like if we take an example of achondroplasia, so achondroplasia is an autosomal dominant disorder. So if 100 patients are detected with achondroplasia, out of which only 80 patients, just as an example, will clinically present with achondroplasia, rest 20% won't show the characteristic manifestation of the achondroplasia. That is why the penetrance of the achondroplasia will be 80%. It's just as an example to explain that it is incompletely penetrance. Now, the skip generation is absent. This is all we know. We have read it in class 11th and 12th. So, there is skip generations are absent because it's an autosomal dominant. Just remember it this way. Each generation will carry on the defective gene and will be expressed without exception. That's true. Otherwise, although the clinical manifestation may vary from individual to individual. Now, the genotype that are present that can be shown in autosomal dominant disorder is either the heterozygous state or the small type, the recessive homozygous, not the dominant homozygous, because the dominant homozygous will die, right? And the heterozygous one will be the affected one, and the homozygous recessive one will be normal. Now, it's just a small pedigree to express how this actually goes. If a normal person marries an affected person, so the normal person will carry the homozygous recessive type and the affected person will have heterozygous type. So their offsprings may have 50% chances to be affected and 50% chance to be normal. So it's a case of 50-50. Yeah. Now moving on to the next slide. Here, now here are the examples. Okay, for whatever I have told you till now, just remember the examples are the most important thing that you have to remember, and you have to remember, no other choice. And here's a little mnemonic for it, like he has a very dominating father or dominant father. So in this he, for H, it stands for hereditary spherocytosis. Don't mistaken it with hereditary elliptocytosis. It's not been included here. Just the hereditary spherocytosis is been included in this. Then another H stands for Huntington's disease or Huntington-Cori. That's the same. You will again hear this term Huntington's disease or Huntington-Cori in the subsequent in the X-linked recessive disorder. So don't get confused. It is present in both of the type, and I have a doubt regarding it as well. I'll actually update an edit on it. Now from A, it is adult onset polycystic kidney diseases. Adult onset polycystic kidney diseases. Very V stands for VHL and von Willebrand disease. VHL, I'm not sure about the full form, but I will make it clear. Then von Willebrand disease. Now from D, the dominant D stands for dystrophia myotonica. Dystrophia myotonica. And O stands for osteogenesis imperfecta. Osteogenesis imperfecta, that is the bone which is formed, which is generated, is imperfect, sort of. Next, the father F stands for familial hypercholesterolemia. Familial hypercholesterolemia. And there are more, like Marfan syndrome. These are the other more examples. You can just remember. It's like Minent. That is Marfan syndrome, intermittent porphyria, neurofibromatosis 1, achondroplasia, neurofibromatosis 2, and tuberous sclerosis. That's all. Marfan syndrome, intermittent porphyria, neurofibromatosis 1, achondroplasia, neurofibromatosis 2, tuberculous sclerosis. Now moving on to the next inheritance type, that is autosomal recessive. Again, the frequency of occurrence will both be same in male and female. Now can only be expressed in homozygous state. Autosomal recessive disorder can only be expressed in the homozygous state. In only dominant type, homozygous dominant type, it will be expressed, not in homozygous recessive type. It won't show the character. And when the person is heterozygous, it may be carrier. It will be a carrier or a trait. So it won't show the features, the manifestation of the disease, but may carry the defective gene. Also, usually it is due to the enzyme deficiency. Right when I tell you the autosomal dominant type, it was due to the structural protein defect in the structural protein, but here it is due to defect in the enzyme deficiency. This is due to enzyme deficiency or inborn errors. Of course, enzyme deficiencies may occur from the prenatal state, antenatal state. Now, skip generation is present. Of course, it is a recessive disorder, so skip generation will be present. People can be carrier, as there was no carrier. The basic difference you can actually get it through. Now, usually manifested in childhood. In dominant type, it was adulthood, and here it is from childhood, and it will be severe. Now, there is complete penetrance. This is the important feature I told you. In autosomal dominant type, there was incomplete penetrance, but in autosomal recessive, it is complete penetrance. Means if 100 patients have been diagnosed with any sort of defective gene and autosomal recessive disorder, so all the 100 patients will have the clinical manifestation. Now, the genotype that will have in autosomal recessive will be dominant homozygous, that is affected. Recessive homozygous will be normal, and the carrier type heterozygous will be carrier or trait. Here's the pedigree. Again, if the affected person marries a normal person, so the chances of their offsprings will be only the carrier. There will be 0% chance to be affected and 0% chance to be normal. They would all be carriers. Moving on to the next video, next slide. Another pedigree. If a carrier marries to a carrier, there will be 25% chances to have the disease to be affected, and 50% chance to be carrier, and 25% chance to be normal. Now here's the example. I have make it a mnemonic like A B C D E F G H I till I. So from A, it is alpha 1 antitrypsin deficiency, ataxia or L-captonuria. From B, it is beta thalassemia, and just always remember that when beta thalassemia, we talk of beta thalassemia, it's a hot topic. You will actually count on sickle cell anemia as well. Now, the next step, that is cystic fibrosis. From C, it is cystic fibrosis or congenital adrenal hyperplasia. Let me zoom it for you. For A, it was alpha 1 antitrypsin deficiency, ataxia or L-captonuria. From B, it is beta thalassemia and sickle cell anemia, structural defect, although don't remember this. Next, from C, it is cystic fibrosis or congenital adrenal hyperplasia. Next, from D, it is deafness. E is emphysema. F is Friedreich and ataxia, ataxia. H for Gaucher's disease and glycogen storage disorder. Gaucher's disease, it's a hot topic again. Next, that is hemochromatosis or homocystinuria. I from it is for inborn errors of metabolism. Next, the third type of inheritance, that is X-linked recessive. Again, it's an important type. The frequency of occurrence, because it's X-linked related, so it is more occur in male as compared to female, as females being the carrier. Now, it is not mandatory that female will always remain the carrier. Females can also be affected, when only in a case when there is random X in as we all know that females have 2 X chromosome. So if randomly one of the X chromosome gets unaffected during the Lyon's hypothesis, so it can lead to the affected female as well. Now examples important. This is Lady Heritage College girls don't care about foolish words. This is a mnemonic. Lady Heritage College girls don't care about foolish words. From L, that is Leishman syndrome. H for hemophilia A and B, along with Huntington's disease. Again, I told you this earlier. In autosomal recessive, you will hear, in autosomal dominant, you will hear Huntington's disease, Hunter's disease, and here it is Hunter's disease. There is Huntington's disease. Don't make it. Don't confuse it with others. Okay. From C, this is color blindness. These two we have already read from our 12th grade, so it will be easier to remember. Next, from girl is G6PD deficiency, enzyme deficiency. And from D, it is Duncan's muscular dystrophy. C for it is chronic granulomatous disease. From A, it is A gamma globinulemia. F for it is Fragile X syndrome, and Fragile X syndrome, of course, is related to the X-linked recessive disorder. W, it's for Wiskott-Albert syndrome. These two are immunodeficiency disorders. These two, the Fabers and Fragile X syndrome, and Wiskott-Albert syndrome. Now, moving on to the next slide. Now, as you can see here, if a normal female marries a normal male, so the females will be carrier and the males will be normal. Here's the pedigree I have made. So just to make it clear, we are considering that female is a carrier and male is normal. So if female is the carrier, so all the 50% of the son will be affected, and the 50% of the son will be normal, and 50% of the daughters will be normal, and 50% of the daughters will be carrier. Now, why did we change from children to sons and daughters, since it's an X-linked disorder, so we can actually differentiate between the male and the female child. But since it was an autosomal disorder, both autosomal dominant and recessive, so we cannot differentiate between male and female for the disorder. So there we were talking of children, but here we are talking of sons and daughters. So make it clear, 50% of the son will be affected, 50% of the son will be normal, 50% of the daughter will be normal, and 50% of the daughter will be carrier, like this. So, or if we talk of in terms of children, so we will say that 25% of the children are affected, 25% of the children are carrier, and 50% of the children are normal, as you can see. Now, moving on another case, if the female is normal, here the female was carrier and the male was normal, here the female is normal and male is affected. So 100% son will be normal, 100% son will be normal because the mother is normal, 100% daughters will be carrier. As simple as that. And if we talk it in terms of children, 50% children will be carrier and 50% children will be normal. Now, moving on to the fourth type of inheritance, that is X-linked dominant disorder. It is a rare type of disorder, X-linked dominant, rarely heard. And in this, the heterozygous state can be present, but there is no carrier. The heterozygous case will be present and there will be no carrier. And the dominant type, the homozygous dominant will be dead. So they won't be affected. So again, the male will be more affected as compared to female because it's X-linked disorder. Now, the mnemonic is Ravi, the shortest mnemonic ever. All we have read for now was so long. This is Rett syndrome, A for Alpert syndrome, vitamin D resistance ricket, and incontinentia pigmentia. The genotype will be, if the female is normal, we have made a pedigree, and the male is affected. So here you will see the 100% daughters will be affected. The female is normal, male is affected, and there is 100% daughters which are affected and 100% sons which are normal. Now, as the father actually give this X to their daughter, 50% child are affected and 50% child are normal, just like that. I hope you like the video. Thank you.